Right. In this session, we will discuss about the internal structure of monocot root and TS of monocot root. TS means transfer section of monocot root. In this diagram, anatomy of monocot root. This topic is related to the anatomy of angiosperms. Anatomy of angiosperms. In this TS of monocot root, it consists of three main regions. The first one is epidermis. First one is epidermis. This is epidermis. The second region in the monocot root, cortex. This is cortex region. In this cortex region, again divided into the three subregions. First one is exodermis, second one is general cortex, third one is endodermis. So these three regions under the cortex. The cortex is present between the epidermis and stele. Next, the central region is called, the central region of monocot root is called stele. The stele is the differentiated into the again three regions. First one is pericycle. Second one is vascular bundles, xylem and phloem. The third region is medulla. So that is three regions in the stele. This is a stele region. It is the central region of the monocot root. So in this in a monocot root differentiated into the three regions: epidermis, cortex and stele. So this is the structure of the internal structure of the monocot root. Internal structure of monocot root. Right. You know what is the example? What are the examples of monocot roots? That is monocotyledon plants. Examples: zea maize, rice and wheat plants. So these all are belongs to the monocotyledon roots. So in a structure of monocot root, it shows three main regions: epidermis, cortex and stelar region. Stelar region. Now what is the first region? Epidermis. Epidermis is the outermost layer of the root. It consisting of tubular or rectangular region, rectangular cells without intracellular spaces. Without intracellular spaces. The epidermis is outermost layer of the thin-walled rectangular cells which are arranged compactly without intracellular spaces. There is no intracellular spaces between the epidermis layer. The cuticle and stomata are absent in the monocot root. In a dicot root, the cuticle layer is present on the surface of the epidermis. The stomata are also present. Okay. Now, some epidermis cells shows tubular extension of the cell wall forming a root hairs. These cells are called trichoblasts or piliferous cells. This is the most important concept, that is forming root hairs. The root hairs are forming from the epidermal cells. Some cells shows the tubular extensions. That is that tubular extensions are forming the root hairs. Next, such epidermis, that epidermis is called epiblemma or rhizodermis. See here, the epiblemma means which cells are forming the root hairs, that cells are called epiblemma. Such epidermal cells are called epiblemma or rhizodermis or filiferous layer. What are the main functions of root hairs? The root hairs are useful in the absorption of water from the soil. And what is importance of the epidermis? The epidermis helps in the protecting inner tissues. So this is the outermost region of the monocot root. So next, the second region in the monocot root, cortex. The cortex is present between the epidermis and the stellar region and more extensive than that of the dicot root. It is a parenchymatous cells and clearly divisible into the hexodermis, cortex, general means general cortex, and endodermis. The cortex is the divisible into the three regions again, that is exodermis, general cortex, and endodermis. So what is the exodermis? It is the outer region of cortex. It is the outer region of cortex and consists of one to two layers of the thick walled living cells with suberin in the mature region of the root, that is the exodermis. The ectodermis, the outermost region of the cortex, the cells are made up of suberin cells in a mature region, that is hexodermis. So what is the importance of the exodermis? It gives a mechanical support, mechanical strength to the root, that is the exodermis. Where is the exodermis? Below the epidermis. The exodermis is outer region of the cortex. And the second region in the cortex, general cortex. The general cortex, this is the general cortex. The general cortex, it is present between the exodermis and epidermis, exodermis and endodermis. It between the exodermis and endodermis. It is a multi-layered extensive parenchymatous with prominent intracellular spaces. So the intracellular spaces are present in the cortex. There is no intracellular spaces in epidermis. The intracellular spaces are present in the general cortex. What is the importance of the general cortex? The general cortex cells are made up of parenchymatous cells. Now each and every cell of the parenchymatous cells contains leucoplast. That leucoplast cells store the reserve food materials. That is the main main function of general cortex. The general cortex is store the food materials. And what is the importance of what is the functions of exodermis? It gives a mechanical support to the root. Okay, what is the main functions of epidermis? It protecting inner tissues. And what is the main functions of root hairs? It absorbs water from the soil. So this is the second region, that is the cortex. Next region in the cortex, endodermis. The endodermis, it is the innermost layer of the cortex that encircle the stellar region. It has compactly arranged barrel-shaped cells without intracellular spaces. The innermost layer of the cortex, the composed of single-layered and barrel-shaped cells that are arranged compactly without intracellular spaces. The radial walls and transverse walls are lignosuberin thickenings. This is the most important concept. Radial walls of the endodermis, transverse walls of the endodermis has lignosuberin cells, or lignosuberin thickenings are called Casparian thickenings. The Casparian thickenings are present in the endodermis. This is the most important concept. So, where is located Casparian thickenings in a monocot root as well as dicot root means that is endodermis. The endodermis is innermost layer of the cortex. Some endodermal cells are situated opposite to the protoxylem elements, or thin-walled and without Casparian bands. The cells are called passage cells. What is the importance of the passage cells? The passage cells allows the water from the cortex to stellar region. Stellar region. Where is the passage cells? The passage cells are opposite to the protoxylem of xylem elements. So, in endodermal cells, all endodermal cells are having Casparian thickenings, but where the cells are present opposite to the protoxylem cells, that cells they do not have any Casparian thickenings. That's why that cells are called passage cells. The passage cells allows the water movement by the symplastic movement. Symplast movement, transportation of water. That is the importance of functions of passage cells. The passage cells helps in the entry of water from the cortex into the stellar region. Okay. This is the second region in the monocot root. In the monocot root, the that is cortex region having three sub regions are there: exodermis, general cortex, and endodermis. So, you have to remember in endodermis the Casparian thickenings are present. Casparian thickenings are present. So, this is the endodermis. Next, the central region of the monocot root, that is a stellar region. It is the central region of the root with the vascular tissue surrounded by endodermis. It is smaller than the cortex. It consists of pericycle, vascular bundles, and medulla. Again, the stellar region in monocot root differentiate into the three regions. Again, three regions: pericycle, vascular bundles, and medulla. So, in this stellar region, the first part in the stellar region, that is a pericycle. It is usually single layered cells and has compactly arranged smaller rectangular cells, thin walled parenchyma cells with dividing ability. Means here the most important concept in a pericycle, in monocot root there is no origination of lateral roots, but in a dicot root the lateral roots are originate from the pericycle. That is the most important concept. In a monocot root there is no the formation of lateral roots. Why? Because that monocot root, the root system is fibrous root system is there. So, pericycle, in a dicot root the lateral roots originate from the pericycle, and that parenchyma cells having dividing ability because the meristematic tissue is there. It gives rise to lateral roots in the maturation region. In maturation region, the lateral roots are formed. Okay, that is the pericycle. It is the first region in a stellar region. The second part in the stellar region, that is the vascular bundles. This is the most important concept in a monocot root. Vascular bundles. Vascular bundles. Vascular bundles. Which type of vascular bundles are present in the monocot root? First of all, we have to collect the information from dicot root. In a dicot root, the vascular bundles are tetrarch condition, means 4 xylems and 4 phloem bundles are there. That is called tetrarch condition. But in monocot root, the xylem and phloem strands are present separately on different radius and alternating with one another at the periphery of the stele. The vascular bundles are radial, separated, and closed vascular bundles. So what is that is the protoxylem in at the periphery and metaxylem towards the medulla. Hence, this type of xylem bundles are called exarch condition. This is the most important concept. What is the exarch? The exarch is nothing but protoxylem is at the periphery, means it is towards the epidermis, and the metaxylem is towards the medulla. Hence, this xylem is described as a exarch condition. Means the vascular bundles are arranged in the alternative. Alternate means that is radial, separate to each other. That is the type of vascular bundles. So finally the conclusion in the vascular bundles in monocot root, the vascular bundles are described as radial, separate, closed vascular bundles with exarch condition. Right. Which type of vascular bundles are present in monocot root? That is vascular bundles are radial, separate and closed vascular bundles. Why we are calling as a closed vascular bundles? They do not have cambium between the xylem and phloem. That is the hence these vascular bundles are described as a radial, separate and closed vascular bundles. Closed vascular bundles. Okay, that is in a protoxylem, the protoxylem is towards epidermis and the metaxylem towards the medulla. Hence, these xylem is described as exarch condition. In a dicot root is also exarch condition. Same, that is protoxylem is towards epidermis and the metaxylem is towards medulla. Hence, these vascular bundles are described as exarch. Usually, monocot root has more than 6 vascular bundles with xylem strands alternating with the phloem strands. Such condition is called polyarch condition. This is the most important identification of monocot root, that is a polyarch condition. In a dicot root, tetrarch condition, 4 xylem bundles and 4 phloem bundles alternate to each other, same radius. So that is a tetrarch condition in a dicot root, polyarch condition in a monocot root. So this is the vascular bundles. What is the main conclusion about the vascular bundles? The vascular bundles are radial, separate, closed vascular bundles. The xylem is exarch, polyarch condition. The usually monocot root has more than 6 vascular bundles with xylem strands alternating with the phloem strands. Such a condition is called polyarch condition. The xylem and the phloem elements are connected by the conjunctive parenchyma present between the xylem and the phloem. It stores the food materials as well as some members of the monocot root become the sclerenchymatous at the maturity stage. So that is the central region of the monocot root, that is vascular bundles. Next, the central portion of the monocot root occupy that is the medulla. The medulla, it is present at the center of the stele or region surrounded by the vascular tissue. It is the extensive and parenchymatous tissue and store the food materials and water. It can store the water as well as store the food materials. Okay. So this is the medulla. The medulla is the central occupy the central place of the monocot root, that is medulla. It is made up of parenchymatous tissue. The final conclusion about monocot root: the monocot root is differentiated into the see in this flow chart we have to remember simplified flow chart about monocot root. The